It was a strange, dark, and eerie night. There was fog all over the ground, and Detective Parker stood there in awe. He was confused. He had just come across a case where colors had gone missing. There were reds, greens, and blues on his corkboard tied together with string, and all he could wonder was who or what had done this. It was a case like no other he had seen. Colors had just vanished. Witnesses were left confused, and there were scared victims, and they were unable to tell red from green and blue from black. There were no fingerprints, no witnesses, and just one question: what or who could have possibly done this? He stood there, sipping his coffee, trying to understand. And then he remembered. He had a lead. There was a scientist, Dr. Evelyn Carter. She knew more about these type of unique cases than anyone else. After spending countless hours and running multiple tests, he was truly unable to understand what had happened. Even after running some special colorblindness tests, he couldn't understand on how this could have happened. That's why he decided to go to Dr. Evelyn Carter.
He enters a laboratory where Dr. Carter is examining DNA samples on a holographic screen. Dr. Carter, I need answers, he says. Colors are rapidly disappearing, and I think it's genetic. What's happening? Dr. Carter says, what you're dealing with isn't a crime. It's a mutation, a genetic condition passed down for generations, known as color blindness. So you're saying someone's DNA is sabotaging their vision? Exactly, Dr. Carter says. A mutation is a change in DNA that alters how proteins functions. Some mutations aren't harmful, like birthmarks, but others can be harmful and affect functions of the body, like how this one affects our ability to see colors. The culprit is a mutation in the OPN1LW and the OPN1MW genes, the ones that are actually responsible for color perception. Normally, cone cells detect colors and send signals to the brain, but when this mutation strikes, those signals are distorted. This is due to a mutation known as a missense mutation. This occurs when the amino acid is changed and it leads to a different amino acid. This is seen with the original vision, where there is glutamic acid, and it becomes valine with the red green color deficiency and the color blindness vision.
Dr. Carter says the crime starts at the molecular level. Let me walk you through it. Inside the nucleus, RNA polymerase is like a detective scanning the DNA crime scene. It makes a copy known as messenger RNA or mRNA, which carries the genetic message out of the nucleus to the ribosome region. During transcription, one of the two DNA strands serves as the template for RNA synthesis. The other DNA strand, not used as the template during transcription, is called the coding strand because its sequence is identical to the RNA molecule produced, with the thymine replaced by the uracil. Two major enzymes used in this process are helicase. Helicase is used to break down the hydrogen bonds and unzip the double helix formation of the DNA. Additionally, RNA polymerase is an enzyme that matches the mRNA or messenger RNA to the complementary bases. For example, it matches adenine to thymine and cytosine to glycine.
So where does it go wrong? He says. Dr. Carter says the problem is in the next step. During translation, the cell uses the genetic information carried in messenger RNA to create proteins, essentially decoding mRNA into a sequence of amino acids that make up a peptide chain. Here, tRNA or transfer RNA acts like a carrier by delivering the correct amino acids. They match their anticodon to the correct mRNA codon, ensuring that everything is in order. Normally, this process works flawlessly by building photopigments that allow us to see the color we do in our everyday lives. However, Detective Parker says, let me guess, one mistake and the whole system is thrown off? Carter responds with, exactly. And in our case, a mutation replaces the correct amino acid with the wrong one, also known as missense. And the result of this? A faulty protein. And without the right protein, The victim or our color-blinded individual loses their ability to distinguish between certain colors. Also, during translation, the mRNA is sent to the ribosome, where it translates the genetic code into photopigments, or the proteins in cone cells that actually detect light. Think of it like a secret code. Adenine always pairs with uracil, and cytosine always pairs with glycine. It's like a precise system. And what happens when the system fails? Detective Carter says. Dr. Carter responds with, well, in colorblindness, the mutation corrupts the code. The ribosome either inserts the wrong amino acid or stops too soon. This means that the protein can either be defective or just missing entirely. This could ultimately lead to the crucial protein of opsine to not be made at all. And Detective Parker says, and without that protein, the victim just loses their sense of color? Exactly, Dr. Carter replies with. Their cone cells malfunction, and their colors start to become blended. Reds and greens start to become indistinguishable, and blues and purples start to become unrecognizable. It's like their world is being rewritten entirely, but in grayscale.
As Detective Parker began to pin a new suspect onto his corkboard titled Genetics, he began to wonder, so, who could be behind this? A mastermind? Or just a single gene gone rogue? Dr. Carter said, the real suspect is inheritance. Most cases of colorblindness are passed down through the X chromosome. Since men only have one X chromosome, if they inherit a faulty gene, they're out of luck and they're stuck with colorblindness. Women have two X chromosomes, so they often compensate with the second one. Detective Parker pondered, so that's why 1 in 12 men are colorblind, but only 1 in 200 women are colorblind. Dr. Carter said, bingo. And sometimes it's not even genetic. Brain injuries, chemical exposures, or diseases can cause colorblindness and other mutations. And these ones can even get worse over time.
Detective Parker says, so let me get this straight. The crime was genetic, the weapon was actually a mutated gene, the victim were not one person but millions of people who were robbed of color in their day to day lives? Exactly, Dr. Carter says. But there is still hope. Special color filtering glasses can help some people see color. And researchers are currently working on a gene therapy, which is a way to fix the mutation at its source. Detective Parker replies with, so one day we could bring color back for those millions of victims around the world? Dr. Carter says, it's possible. This case is not closed yet. This case, it's different. No crime, no fingerprints. It's just biology gone rogue. Some mysteries aren't solved with handcuffs, they're solved with science.